What if the biggest bottleneck in understanding health and disease and finding new cures wasn't just our ability to see the impossibly small, but the overwhelming challenge of knowing where to look and what to focus on amidst trillions of cellular events?
We suddenly started looking not on 25 cells, but 10,000.
There were clear drawbacks in the technologies that we were using.
But this quest has been incredibly challenging. Imagine trying to find one specific grain of sand on a vast, constantly shifting beach. That's the scale of the problem. Finding that one critical event among millions is like searching for a needle in a cosmic haystack, a haystack that's alive and constantly moving.
From the perspective of an experimentalist, I've been sitting at the microscope with the sample that will only be good for, let's say, an hour. And it means that in an hour, realistically, maybe you only find 3 or 4 events that you will image, and then you will never know, basically, how the rest of the sample looked like.
Each lab becomes so specialized in what they do that others will have a difficult time reproducing it just because it's technically difficult.
This painstaking search is often tedious, incredibly time-consuming, and worse, it's plagued by an invisible barrier: human bias. We're all wired to see patterns, to look for what we expect. But in science, the greatest discoveries often lie in the unexpected, the anomalies that our focused attention might miss.
Before, I would just basically spend the same amount of time by the microscope, looking at what is interesting to us, and most often than not finding anything.
This limitation means countless hours, sometimes entire careers, can be spent yielding just a few observations, capturing only a fraction of the story. Discoveries are delayed, vital medical advancements slowed, and the very nature of this specialized manual work makes it hard for other labs to reproduce the findings.
Core problem that we're trying to solve was always there. To capture images of events in the microscope from a purely data-centric way. No human intervention in the loop, it should all be automated.
What if, instead of microscopes being passive windows, we could teach them to become active participants in discovery, to actively understand what they're seeing and guide the search objectively and at scale?
Okay, so I've just put my sample into the microscope. It's a sample of mouse keratinocytes. I'm just going to close here. So what I want to do here is to measure the intensity of the mitochondria in the different cells. So to do that, I'm going to start a bit larger sample scan to get enough cells to have quantitative data. And then I'm going to use Cytely to analyze and get the job done, pretty much, you could say. So now I'm going to, first of all, make a new experiment in Cytely. I'm going to select the correct camera and the correct microscope here. So I decided to make a scan of approximately 16 images, which is not super crazy, but it should work well for me. So what happens now is that the microscope starts taking images. Hopefully they're going to be like in focus and nice.
This fundamental challenge and the limitations of traditional methods spurred efforts to develop a new paradigm, data-driven microscopy. It's an approach that's transforming the very lens through which we view life, making the acquisition of quantitative, high-fidelity microscopy data much more accessible and powerful. So how does this new approach really work in practice, and what makes it different? Here, through Cytely, the company founded to bring this technology to researchers worldwide, we get a deeper look into this process. The process begins with a rapid automated survey of the entire sample. The system creates a complete digital map. Instead of a researcher manually searching, the software systematically captures an overview. And this is often done at a lower resolution to be fast and gentle on the sample, especially if it's live. From this initial map, the system automatically identifies every single cell, measuring and quantifying its characteristics. This is crucial. It's not just about collecting the images, it's about instantly turning those images into rich, analyzable data, quantified, objective data. Researchers can see patterns across thousands of cells this way, and not just a handful. And here's where the smart aspect becomes really powerful. Based on the data from this comprehensive scan, the researcher can now instruct the system to find and zoom in on very specific cells or events of interest. This means filtering out the statistical noise and focusing only on what truly matters for the research question, all without human bias, manual searching, or guesswork. The microscope is then automatically directed to revisit only those pre-selected high-value targets for detailed high-resolution imaging. This targeted approach saves enormous amounts of time and data storage, and crucially for live samples, it minimizes damage by significantly reducing overall light exposure, leading to more physiologically relevant results.
Each lab becomes so specialized in what they do that others will have a difficult time reproducing it just because it's technically difficult. That is well known from other fields as well. It's something you have to consider, but it's also hard because you put yourself in a difficult position because more work, it's more rigorous, and it will take perhaps more time to get to the results.
This ability to automate the tedious, to let data guide discovery, and to focus human expertise on analysis rather than manual labor isn't just an incremental improvement. For many researchers, it's a revelation, fundamentally changing their relationship with their work and the very questions that they even dared to ask.
So if we can shift that and make microscopy much faster, more efficient, as we are doing now with smart microscopy approaches such as DDM, I think that will just elevate science in general. So we will make discoveries faster, we will develop drugs faster, because it will be quicker to rule out the bad sort of outcomes and be quicker to find the good ones.
The impact of this new way of working is significant. Early applications of data-driven microscopy demonstrated the potential for a 1,000-fold increase in data throughput and insights accelerated by 60 times. One of the foundational academic explorations of this method even highlighted a saving of what amounted to 50 months of manual labor in a single automated run. This efficiency doesn't just save time, it frees researchers from the practical limitations of older techniques, allowing them to pursue more ambitious and truly groundbreaking questions.
Data-driven microscopy is a term that we introduced. Perhaps others probably thought of the term as well before that, so we wanted to just make some simple things to start with, to capture simple events or so. But then eventually that evolved, and we realized that we could actually build a much more advanced and general way of doing this that we can then apply to all our scientific questions.
And seeing 10,000 cells instead of 25 isn't just about bigger numbers. It's about capturing the true diversity of life at a cellular level, finding that one rare cell behaving differently that might hold the key to a new drug target, or understanding how an entire population truly responds to a treatment, not just a few convenient, potentially biased examples.
I think the turning point for me when I truly understood the power of data-driven microscopy. The moment it changed for me was when I was collecting data for one of the figures. I set up the experiment. The task was to find these different types of cells in my sample. And for one of the types, it was below, I think there was in total 200 of these cells in this sample overall. I set up the experiment, I pressed play, I went home. And then in the morning when I got back, I had captured 16,000 images. Among these were the type of cell that I was interested in. And I was just thinking to myself that, okay, if I captured these images manually, 1,600 images, it would take me, I don't know how long, weeks. And I slept. I was at home sleeping. It was the best time ever.
I'm very curiosity-driven in terms of the research, and I get frustrated when I don't have the technology to answer the questions that I ask.
One of the most important things was definitely getting the ability to see what is happening on the full cell population level. We suddenly started looking not on 25 cells, but 10,000. And that really changed how we approach the things. It also impacted the way we were performing the experiment itself. It actually showed us that the approach that we had from the very beginning was wrong, and we needed to change things in order to actually make the experiment work. In that way, it was very transformative.
This power isn't limited to just one field. In cell biology, researchers track cell division with unprecedented precision. In drug discovery, responses to compounds are screened automatically at scale. In neuroscience, neural activity is monitored in real time. In developmental biology, growing structures are followed across generations. It's about uncovering those critical events at the micro and nano level that previously slipped by unnoticed, accelerating research across the board. A significant part of this future lies in the integration of artificial intelligence.
If we have more smart microscopy approaches, and especially if we also have the metadata that is possible through these automated approaches, if we collect the right amount and the right type of metadata, that will then be an enormous valuable resource for AI to go in and make connections that we can't see as easily as humans.
Perhaps we can use advanced AI-based tools to really recognize and get that level of information without using the approaches.
So I think one of the big ideas that has been around and that I'm sure will be soon implemented or already implemented in some ways is to use large language models to really allow the user to talk to the microscope and plan the experiment. So you don't need to know actually any programming or any thing like this, no scripting, but you basically can tell the microscope what you actually want it to do and it will design your experiment for you.
The ultimate vision: to democratize this sophisticated technology, removing bottlenecks so labs everywhere, regardless of size or specialization, can unlock insights hidden in the microscopic realm, making breakthroughs happen faster.
Imaging-based diagnostics is essentially required worldwide for a wide variety of problems. If you're not excited about the fact that people in any remote area of the world can take an image, and nowadays you can take an image with your cell phone or through a small microscopy attachment to a cell phone, and then use really advanced microscopy tools to real-time diagnose or figure out what you need to do next. I mean, who is not going to be excited about this?
Perhaps even using this deep understanding to help design biological materials for new functions, fighting disease or cleaning our environment. But that's the story for another time. What's undeniable is that this convergence of smart automation, data science, and artificial intelligence isn't just improving microscopy, it's fundamentally redefining its potential and role in biomedical research. I believe Cytely ultimately will positively impact millions of lives thanks to the hardworking scientists we have the privilege of serving. AI is not just changing how we see the microscopic world, it's changing what we can discover. By turning data into insights, we're moving beyond observation to true understanding. The era of purely image-driven microscopy is ending. The future is data-driven, intelligent, and brimming with potential. Together, we are taking microscopy from an image-driven art to a data-driven science. This revolution in how we explore the microscopic realm is more than just a technological leap for biology, it's a glimpse into a future where the fusion of intelligent automation and human ingenuity accelerates discovery across all scientific frontiers, empowering us to understand our world and ourselves like never before.